Hello guys and welcome back to Excel Share Academy. So today we are gonna do the last topic in the nucleus. Okay. So before I proceed, so there was this question which I have asked yesterday regarding that. This was asked in the interview, and if you remember, I have asked this question where they were asking about the radius is given and how many integral proteins are present, right? So Shruti, would you like to take this up and try to explain? Did you write this thing off from somewhere you got?
The formulas and everything written I wrote. The two diagrams on the left are taken from the internet. The right I just constructed.
Acha. No, good. So I'll just repeat the question and then you can explain like how you got this answer. Okay. So the question was, so we have got a nucleus and it has got the radius of around 10, okay, 10 micrometer. And there are integral proteins, or let us say NPCs are present, whatever it is, okay. Now, it was given that there are 1200 NPCs are there, okay. So they might replace NPC with some integral proteins, or they might say something else. Let it be anything else, anything. Then the question was, what is the distance between two NPC? Okay. Yes, Shruti, over to you. How do you explain this?
Yeah, so there were a couple of assumptions that we made. One was that the nucleus would be a perfect sphere. So for that, since the NPCs would be on the surface, I first figured out how much surface area we have. So surface area for a sphere was, yeah, so we were just taking it as if there is nothing biological over it. We're just dealing with spheres and things that are supposed to be on the sphere. So surface area of the sphere was 4 pi r square. So r would be the radius of that sphere. So in this case, it would be 10 micrometers. So that surface area is there. Now, if you divide that by the amount of things that are supposed to be distributed on it, then you will get squares of the entire sphere by which are equal, what do you call it? Squares of equal size. So all I had to figure out was the distance between the midpoints of the two squares, which would be equal to the side of the square, which is the same as the answer we get. Yes. So that's what I am trying to say in the figure at the rightmost section. So x is equal to one side of the square. X is also equal to the distance between the two midpoints of the square.
Yes, and that's
so
why
if there was a pattern to the nuclear pores, then this answer wouldn't work. But then since the question was specifically the distance between them, this does work.
Yes. So that's why you get around 1.04, or roughly we can say 1 micrometer is the distance between the two NPCs. So I hope Akshaya and Tanvi, did you get the answer?
Yes.
Okay.
The part where it was considered as a square, I don't get that.
Shruti, would you like to explain again? Like how are you taking this part, this figure where you're explaining the square thing? एक बार फिर से समझाओ. See, that's how you're going to learn like how to teach also, right? Yeah.
So actually what I did was sphere ka surface area hai. Now we put it on a flat surface. So now we have something surface area, which is a two-dimensional structure. If you divide it by the number of things, if you take it as distance, since the number of things are, let's say, together, so you will get exactly that distance.
Why it is taken as a point of square, or why not a circle? Because a pore size or nuclear pore complex is kind of cylindrical in shape, and it can be considered as circle, right?
Yeah, that is why I said before that we were making a couple of assumptions. One is that nucleus is a perfect sphere, which I don't know if that is true or not. Second is that the nuclear pore complex will be measured from the midpoint. So I'm not taking into account how big the nuclear pore will be. I'm just taking into account what will be the distance between the midpoints of the two nuclear pores. So I'm not taking it as a three-dimensional structure. I'm just taking it as points which are scattered across a spherical surface.
Because I too did the same thing. Even I got the same answer. So, but the method was a bit different. Yes, so that's why I wanted to
Okay.
clarify.
Okay, so I'll explain this method later. Actually, would you like to try explaining your method? Like you can write somewhere here if you want to share. Like what was your method of solving?
Like I didn't take anything specific in terms of whether it is a circle or anything, ma'am. Just like there is total number of NPCs are 1200. Then the so the distance, I mean the space occupied by the nuclear pore complex is going to be 1.04 micrometer. Uh, I got help from my brother also. Like he said that further you can take so this is so the 1.04 micrometer is nothing but the space occupied by a single nuclear pore complex, whereas the distance between you need to find the distance between two nuclear pore complex. So so we took it as pi r square, that is the area of the circle, that is a nuclear pore complex. So by solving pi r square, so we are going to find out the radius. So and the radius is going to be the r square. So we are going to find a root, and then it is going to be doubled because we need the distance between two circles. So I was getting the answer of 1.15 micrometer. Like further after this.
Wow, wow, wow. This much complications of a simple question.
I did not
No,
understand.
okay, I'll tell both the scenarios, okay? Tanvi, do you have any third method of solving?
No, ma'am. I just directly I divided it by 1200 and got my answer. Like
Oh.
it was a simple. I didn't think that much like Akshay and Shruti. I just directly divided it.
हाँ, so actually exam में interview में you have to go according to what Tanvi did. ठीक है, इतना explanation नहीं चाहिए. But both of you कुछ गलत नहीं है. There's nothing wrong. It just got more complex. ठीक है, and becoming or giving any answer in a complex way, it shows that you're not thorough with the topic. So I'll go one by one. So what Tanvi did, and that is what I will expect from my students. ठीक है, कि अगर मैंने interview में ये पूछा है, so already hint was given that the nucleus is a sphere, okay, and I'm asking, see अभी तो मैंने NPC बोला है, it would have been some other protein, it can be some other protein. Akshaya, your mistake was that who or what, like, or maybe you would have assumed that these proteins are going to be circle in shape, है ना?
On the surface, yes.
हाँ, no, but you cannot, right? When I'm saying that nucleus sphere है, किसी 3D के ऊपर मैं दूसरा 2D क्यों लूंगी? When you talk about protein also, any protein, again, this is going to be 3D only in shape. ठीक है? Are you getting my point over here? That on a 3D, why are you taking 2D? Your protein is also again a 3D. Whether you take NPC or you take any other protein, it is gonna be 3D only. जब आप एक जगह 3D consider कर रहे हो, आपको दूसरी जगह भी 3D ही consider करना होगा. ठीक है? So there was this small mistake, but that's okay. Okay, so let it be anything. Okay, so what usually we do is that आप surface area लो, जो भी आपको value आएगी, just divide by total number of the given proteins will give you the reasonable distance between the points. ये हो गया आपका simple तरीका, ठीक है? Same thing Shruti has done. It's just that in a engineering way. Okay, so she has taken the sphere, she got the surface area. Now she has assumed that the nucleus, not the nucleus, the center, this distance is gonna be same to each NPC, correct? है ना? That is another. This was your
Yeah, I thought
second.
about solid angles first.
हाँ, this is your second assumption. इसकी जरूरत नहीं थी actually, because you know that NPCs equidistant नहीं है from the center of the nucleus. Correct? But since we assumed everything.
You also said about how the NPCs move according to the requirement of the cell, right?
Yes.
So anyway, that is why I was like, actually it won't be like this.
हाँ, so anyways, the first assumption जब हमने बना लिया था that nucleus is a sphere, so it is not a perfect sphere in reality. So when you said about your second assumption, that can also be considered because our first assumption is also not actually true, right? Nucleus is not a perfect sphere. So आपका second assumption भी मैं ले सकती हूँ. Then that the from the center the each NPCs are going to be equidistant. ठीक है? Then you said that let us open up this 3D structure into 2D. Right? That is what you mean. When you open it, it will be something like this.
Yeah.
And when you cut it and then you make the squares, and when we see, when you do the alignment, you see that these points of the NPCs, they are at equidistant. That's how you calculate and you get this thing. So घूम फिर के सभी ने वही आंसर निकाला. It just that 1.04, 1.15 या 1.06. It's again based on the pi value. आपने 3.14 लिया या 22/7 लिया. It also matters. But of course you got like it is around 1 micrometer equidistance. It is present. Okay. Is it clear, Akshaya? Like how Shruti was doing?
Yes, ma'am. Got it.
Okay. Okay. Shruti, did you get how Akshaya was doing?
Not exactly.
So instead of considering here, she assumed that these all to be circle, right? Right, Akshaya?
Yes, ma'am.
So here instead of so
like I got 1.04. I got that it is 1.04. I further complicated it, ma'am. Like
yes, by
finding distance between two.
Yes, by taking the area, then taking the root of the R and all, right?
Yes. Yes.
So that was not required. That's okay.
Achha, so she tried to exclude the NPC
So
area
she
to find out the distance between.
So she consider NPC as circle and uska फिर area, फिर uska root R वगैरह वगैरह.
Okay.
So I really believe that this exercise was fruitful. You guys have taken effort. It was a good discussion also. ठीक है? This is what I always wanted. ठीक है? मुझे नहीं चाहिए कि मैं हमेशा ये बोलूं श्रुति तुम बताओ, अक्षय तुम बताओ, तन्वी तुम बताओ. I want the response coming from your side. I'm telling you guys, offline होता ना, तुम लोग मेरा क्लास आना छोड़ देते because I'm a very strict teacher. It's just that online I'm not able to do anything. चलो, anyways. It just so little busy also going. I know I can see Akshaya your questions are there. I have gone through. It's simple also. It's just that I just need time, and maybe Friday I'll be free. After Friday I'll be free. So Saturday Sunday definitely we'll have the session. Okay, question answers. Just remind me once so that we can have this discussion. Okay, so let's move on to our topic now. The last topic in the nucleus is to know about the nucleolus and the rRNA. So basically we know what is nucleolus, what is rRNA. So in the nucleolus, the RNAs are getting synthesized, assembled. बस इसी चीज को हम थोड़ा डिटेल में भी पढ़ेंगे, ठीक है? So it is the site of RNA transcription and processing as well as ribosome assembly. Now we are going to see like how exactly the entire ribosome assembly is happening. अभी हमने पढ़ा कैसे है that your 30S, 50S together ऐसे होता है वगैरह वगैरह, right? So how these RNA or the ribosome components are being transported from nucleus to the cytoplasm, that is what we're going to see over here. So the nucleus is a ribosome production factory designed to fulfill the need for regulated and efficient production of RNAs and the assembly of the ribosomal units. So we know all this 5S, 5.8, 18S, 28S RNAs in the case of the eukaryotes. Similarly, we have for the prokaryotes also, right? Now, in the prokaryotes, we don't have any problem. Kyun? Because wahan pe na nucleus hai, ना कोई अलग separation है. So there we don't have this problem of transportation. The transportation problem comes only in the case of eukaryotes, right? So जहाँ पे from the nucleus to cytoplasm we need the transport. Okay, so 5.8, 18S and 28S. Now guys, this is something you have to remember. This is transcribed as a single unit within the nucleus by the RNA polymerase 1, which directly gives you 45S ribosomal precursor RNA. Okay, I'll show the figure. Now this 45S is processed to the 18S RNA of the 40S and to the 5.8 and 20S of your 60. ये आपको मालूम ही होगा. I'm sure you must have known about this. Like 40S में क्या आता है, 60S में क्या आता है, like this. So you can just ignore the prokaryotic 70S ribosome, just concentrate on the 80S. So you know 60 में आता है आपका 28S, 5.8 and 5, whereas in the case of 40 you have got the 18S. Okay. Now suppose again, as I mentioned before, that there are certain universal primers are there. Same way in the case of eukaryotic organisms like yeast ho gaya ya koi cell lines ho gaya, if you don't know what kind of cell it is, okay, and you want to identify, so what we can do is we have got those universal primers which is made, or the primers are based on this 18S RNA. So you get the sequence, okay, you carry out the PCR, you get the sequence from outsourcing from the company, from the sequencer, and you align in the blast. Blast क्या है? Basic local alignment tool. आपने पढ़ा होगा fast, blast in the NCBI. थोड़ा सा bioinformatics पढ़ा होगा, I believe everyone. Blast. Have you heard about the blast tool?
Yes, ma'am.
Yes. So in the blast you have got like for the alignment of protein रहता है, gene रहता है, right? So all this thing you can align it over there. So आप blast में डालोगे and then you can check like how much similarity it is coming to the organism, which organism it is coming. If it is 100%, it means ये वही organism है. 90% है, so it is belonging to the same species या same genus, ऐसा कुछ होगा, ठीक है? Okay. So please do know about all this blast का full form and all everything. आपको थोड़ा मालूम होना चाहिए. It is a basic local alignment search tool. थोड़ा बहुत तो पढ़ा ही होगा, but yes, of course, one more thing is that nobody is asking in the exam. The blast क्या है, FASTA format क्या है, nobody is asking. I'll let you know, like in bioinformatics आपको क्या prepare करना है, ठीक है? Now look at over here. So this is how the transcription of the ribosomal RNA is happening. So you can see that these, you know, these DNA loops out wherever the transcription is required. So as you can see, this is one stretch of DNA and only one promoter is there for your 18S, 5.8S and 28S. So directly एक साथ ये transcribe होगा and then you have got the RNA processing where all these RNAs will be processed and separately assembled to form your smaller subunit of your eukaryotic ribosome. Clear? This I have mentioned UBF, SL1, RNA polymerase 1 in the transcription, if you remember. Anyway, so anyways, everything will be like once you start remembering all this thing, जब आप ये सब याद करने लग जाओगे ना, then you can make a link कि थोड़ा बहुत सेल बायो में पढ़ा है, थोड़ा बहुत हमने मोल बायो में पढ़ा है, ठीक है? Okay. UBF, upstream binding factor, selectivity factor, polymerase 1, all together is helping in the rRNA transcription. Clear? Is this slide clear to everyone? This image is clear to you? This one? If you see, if you remember, आपका DNA chromatin कैसे रहता है? ऐसे से नहीं रहता है, zigzag, zigzag. So they loop out. So wherever those regions are there, that region gets आपका transcribed. Okay? Yes? Doubt है तो अभी पूछो. Akshaya?
Clear, ma'am.
Okay. Now, So this also we have talked about the base modifications in the ribosomal RNAs. So if you remember about the pseudouridine ribose methylation, we talked about when we were talking about the tRNA, uridine, pseudouridine, if you remember, right? So those are these base modifications which is important. As the structures yaad karne ki zarurat nahi, it's okay, you can skip that slide. Now coming to the ribosome assembly, okay? So now what is happening is that, see, the proteins are outside, okay? Ribosomal proteins are outside. So guys, do you remember जब मैंने आपको last time बताया था कि there are certain proteins like CRM1, if you remember. So your RNA along with the CRM1 exportin proteins, it goes outside the NPC and वहां पे फिर आप assemble करते हो and then you bring it back. Do you remember in the case of RNPs? Any little remembrance? No? Okay, just forget then. I'll just repeat once again. Okay. So in that case, what was happening was you are taking the RNA from the nucleus to the cytoplasm, and there those RNA associated proteins were getting assembled, and then that together was recruited inside the nucleus. Okay. Now here, see, you have got, look at the figure. So you have got the DNA here, correct? DNA है, DNA से आपको pre RNA मिल गया. Now the proteins which are associated with the ribosomal RNA, they are outside. So first that will be recruited inside the nucleus. So आपने पहले इनको import किया, ठीक है? Now they go and bind associated regions so that the splicing mechanism will understand where to have a cut or where to have the splice. So you know that cutting of the RNAs. So according to that, then you get your 18S over here and rest over here, and then they assemble together. Then the entire ribosome after assembly of the RNA along with your proteins will be taken outside. Is this image clear? Should I repeat again? Yes. So in the cytoplasm, you have got the assembled 40S and assembled 60S. Now, once your mRNA comes out, then they start assembling and forming the pre-initiation complex. Yes. Any doubts over here? Shruti, would you like to explain?
Okay. So first, since the ribosomal proteins, the ones required for making them are out, they are first transported inside the nucleus, and then they bind and make the pre-RNA, and then they are processed and made into the 18S. Sorry, the 40S and 60S subunit is completely made inside the nucleus, and then the whole protein is transported outside.
It is not a protein, it's a ribosomal unit, subunit, right? 40S, smaller subunit and large subunit. That is transported out. Okay. Okay.
Yeah, the entire thing is out.
Okay, so yeah, so that is about like how nucleolus is performing the function. Now coming to the polycomb bodies, so they are the centers where the repression is happening of your transcription. Repression matlab kya hua? That is inactive regions. Okay, now how this is made inactive, so that is what we are going to see. See, so these polycomb proteins, they are responsible for your transcriptional repression of a variety of genes. How? By methylating your histone proteins. Okay, please do remember. Now don't get confused. So histones get HAT protein and HDAC enzymes. So इनका क्या काम है? Histone acetyl transferase, histone deacetyl transferase, right? So इनका क्या काम होता है? To make your chromatin condense and decondense. ठीक है? So that the DNA replication or transcription can take place. Here, in this case, it is the methylation of the histone at this particular region, H3 lysine 27, where the methyl group is added so that it becomes transcriptionally inactive. Okay, so these are the protein which consists of two complex. One binds to the methylated H3 lysine 27 residues and other is an enzyme which methylates the adjacent histone protein. So as a result, what is happening? They are spreading to the adjacent nucleosomes and generating a repressed chromatin. यानी कि क्या उतना पूरा region आपका inactive हो जाता है. ठीक है? Let's see how these proteins are functioning. So these are your polygon proteins. So it is a repressive complex PRC which binds to the methylated H3 lysine 27 and PRC2 methylates lysine 27 residues of the adjacent nucleosome. ठीक है? सी यहाँ पे देखो PRC1 ने एक को किया है तो PRC2 ने दूसरे को किया है methylation. ठीक है? So eventually what they have done, together this entire complex, the PRC repressive complex, what they have done is this particular chromatin they have made it complete repressive. Okay, now what will happen because of this methylation? Now the histones won't be able to leave the DNA. Now DNA free नहीं होगा तो ना ही आपका transcription होगा ना ही कोई activity होगी. So there will be no gene expression. So now you know the application or the function of the polycomb proteins. Yes, is this part clear? See, PRC2 अगर इसको कर रहा है तो उसके बाजू वाले को PRC1 करेगा. Okay, now this is the same thing. So what they have done is again the immunofluorescence they have done. So it is the analysis. So they have used the green fluorescence protein which is against the polycomb protein. So wherever they bind, it will tell you the regions which has been repressed. Okay. See, हम काफी images देखेंगे and it will all look same. It's just that your antibodies should be specific against which you are studying your protein of interest. Okay. अभी कल को if I show you this, you won't be able to understand whether it is the polycomb proteins binding or it is the apoptosis happening inside a cell or it is what. By just looking at the figure, you won't be able to understand. It is the at that time what antibody you're using, how you're using, that is important, and whether you're able to get the information from the image or not. Is this much clear, guys?
Yes, ma'am.
Okay.
Yes, ma'am.
Guys, please join back. Time is getting over.
Yes, ma'am.
Okay. So these polycomb bodies are frequently associated with the heterochromatin region. So we know like now that heterochromatin region, why it is the inactive region part. Okay. Transcriptionally, it is inactive. So now we know how it is maintained inactive. So if this question comes, your answer should be it is because of the methylation at your histone H3 lysine 27. Okay. This should be the answer. Who does it? It is your PRC. ठीक है? Now, in some cases, polycomb bodies may be formed as a result of the repression of a series of adjacent genes. यानी कि एक जीन यहाँ पे है तो वहां पे आपका polycomb body present रहेगा. It means what? There are PRC present along those nucleosomes. Okay, तो वो polycomb body हो जाएगा. So they are the repressed regions of your chromosomes. So, however, at least some polycomb bodies contain clusters of these repressed domains from different chromosomal regions also. Analogous to transcription factories, they are the clusters of coordinately repressed genes. एक हमने पढ़ा था transcription factory. So if you remember, there were these loops out, DNA loops out, and where the RNA polymerase sits and the other transcription factor sits, and they keep on translating. They keep on doing the transcription, and you get the RNAs, which is then taken outside to the cytoplasm through NPCs, right? If you remember, ऐसे structure था. Okay, so those are known as the transcription factors. Now, in contrast to that, we have also got the polycomb bodies जहाँ पे कुछ भी नहीं होता है. Okay, they are transcriptionally inactive regions. So this is how it will be look like. एकदम आपका एक अलग separate bodies you can see where you have got these polycomb proteins. What are these polycomb proteins? They are your what? Which protein it is? repressive complex, right? PRC1 and 2, which we have discussed. So they keep on methylating your histone proteins so that they are totally repressed. Okay. Now the last things about the Kajal bodies and the speckles. What are these actually? So they are the for processing and the storage of your SNRNPs. Now, where do you find SNRNPs? Yes, Tanvi? I have mentioned this. What is SNRNPs? Full form is small nuclear ribonucleoproteins. Where do we have studied about the SNRNPs? Anyone? So they are the spliceosomes. If you remember in the intron splicing we have discussed. Okay. जहाँ पे U1, U2, U3, 4, 5, 6, there are different types of spliceosome proteins are there which helps in the intron splicing, where they form the lariat formation is there, etc., etc. Okay. So both Kajal bodies and the nuclear speckles, they play important role in the assembly and the storage of your SNRNPs, which is responsible for the splicing. Now, Kajal bodies, they are the small nuclear bodies involved in assembly of SNRNPs and other RNA protein complexes. So they are the first to be exported to the cytoplasm, where they associate with the proteins, they form the SNRNP, and then they come back to the nucleus. Now, why? Because they are the RNA plus protein complex which is helping in the formation of the spliceosome. Spliceosome की जरूरत कहाँ पे है? For the intron splicing. Introns कहाँ present रहेंगे? In your RNA, which is present in the nucleus. Okay. So this is what I explained in the last lecture, where first the RNA goes outside from the nucleus to the cytoplasm, where it is going to get associated with its protein, and entire this SNRNPs will be recruited inside the nucleus. Okay. Now then they are stored in the Kajal bodies. See, this is, I think this is the same structure. So where we have got the CRM1 exporting proteins, it binds to the small nuclear RNA, it goes out, the protein binding association happens, then with the help of this nerpotin, okay, importin, it will come inside of the nucleus. So this is how your SNRNPs are brought inside the nucleus. Is this part clear? Like what Kajal bodies are? Is it clear? Okay. Okay, so this is how your SNRNPs are getting transported. Now, what all is happening in the Kajal bodies? So this includes modification of your small nuclear RNAs by the ribose methylation, that is your pseudo-uridinylation, and the modification of the pre-RNAs. Okay. Now, with the being consistent with the processing of this in Kajal body, the enzyme which is responsible for RNA methylation is the fibrinogen, which is again concentrated in the Kajal bodies. Now don't get confused over here. वहां पे भी आपका methylation हो रहा था, but there the methylation is of your histone H3 proteins. Here it is your RNA which is getting methylated. ठीक है? With the help of the enzyme. And now where it is happening? In the Kajal bodies it is happening. Okay. So these SCA RNAs, that is your Kajal body specific RNAs, they're related to the SNO RNAs in the nucleoli and similarly serve as guide RNAs. Now guys, where have you heard about the guide RNAs? RNA सुना है कभी?
Yes, ma'am.
CRISPR सुना है?
Yes, ma'am.
हाँ. So this CRISPR-Cas, RNA-I, so all these are your RNA interference. CRISPR-Cas हम आजकल use करते हैं to make any transgenic. So there you need the help of the guide RNA so that you can make a specific transgenic mutant or whatever it is. Okay, so all this will be covered in your techniques, don't worry. So what you have to understand here is that they all are like present in your Kajal bodies. ठीक है. ऐसे तो आज तक कभी क्वेश्चन नहीं आया है रिगार्डिंग फ्रॉम द कजाल बॉडीज एंड ऑल. This is how it looks. Again, you have to just, tag the protein which is specific to the Kajal bodies. For example, here it is fibrinogen. So here they have tagged with the antibodies which is specific to the fibrinogen. So you get that fluorescence you are getting. ठीक है. So by looking at the image, you won't understand कि क्या है क्या नहीं चल रहा है. But that's okay. And आज तक एग्जाम में भी नहीं आया है. So if you want, you can skip this part, but this is for your information. So in addition to their role in SNRP assembly, they appear to play a very important role in the telomerase. So if you remember what telomerase does is, wherever you have got the end replication problem, there the telomerase enzyme comes, they use their RNA as template in order to increase the length. Okay, if you remember. So this Kajal bodies, they help or promote the assembly of the RNA protein telomerase, so that they can help in the lengthening of the telomeres. Otherwise, what will happen because of this end replication problem, if suppose telomerase was not there, तो आपके chromosome हर replication round में छोटे होते जाएंगे. Eventually, they will result in the loss of the genes, and that's why you have telomerase in all your stem cells. It is present. Okay. Now, all this assembly of your telomerase happens in the Kajal bodies. Now, so following the assembly and maturation in the Kajal bodies, all these SNRNPs are then transferred to the speckles. Those are your nuclear speckles. So they are again a discrete nuclear bodies, which contains the machinery for your mRNA splicing machinery. So splicing factors are concentrated in the speckles. In short, what is happening? All those your spliceosome machinery जो है आपकी nuclear bodies में रहती है. Nuclear bodies में भी we have got different categories. Just now we have seen Kajal bodies, polycomb bodies, all these are there, right? So this spliceosome is present in your Kajal bodies. Clear? It's okay, you can make short notes also. So already we saw that under the nuclear bodies, we have talked about the nuclear speckles. Okay, nuclear speckles are there. Kajal bodies are there. Then polycomb bodies are there. We have talked about. Okay, so Kajal bodies में आपके telomerase को assemble करने वाले components हैं. Polycomb is meant for keeping your chromatin as transcriptionally inactive region, where we have got the enzymes like PRC1 and 2 doing the methylation. Okay, then what we have discussed, then we have discussed about the spliceosome, which also again happens. Now spliceosome machinery जो भी आपके concentrate होते हैं वो आपके nuclear speckles में आ जाते हैं. ठीक है. So listen to the video again, make short notes. Already one table was there where you can get the list of the nuclear bodies. So this is how it looks. You can see so many concentrated regions are there. So all these speckles carry these splicing factors. Okay. So that's all, guys. With this, I can proudly say that this chapter or this topic is over. And now we will be starting next by tomorrow, the ER. Okay.